For today's topic, we are going to cover the identification of pathogen in bodily fluid. And here's the table of contents for this group. For pre-treatment, there are a few important steps for the collection and transport of blood culture. First, remove the plastic cap and clean the top with 70% alcohol wipe and allow the area to dry. Next, disinfect the puncture site with the new 70% isopropyl wipe, followed by 2% chlorhexidine antiseptic, which is also known as the ChloraPrep. Allow the ChloraPrep to dry completely for at least 30 seconds. Collect the blood using a syringe attached to a butterfly collection system. Attach the syringe to blood transfer device and fill the blood culture bottles with required volumes. The blood culture bottles must be standing upright to ensure that there is no backflow of the bottle contents and appropriate volume of blood is added. Transport bottles to the microbiology lab as soon as possible. When using the pneumatic tube system, make sure adequate foam padding is in the carrier and send no more than 2 bottles per carrier. For the quality assessment of blood culture, the optimal adult fill volume is 10 milliliters, and for children to collect no more than 1% of total blood volume. There is a fill line on the bottle, so underfilling can cause possible false negative results, and overfilling could also cause inaccurate results. The bottle should also be correctly labeled. For the collection of cerebrospinal fluid, it is usually done by trained professionals. The best site for spinal tap is interspace between 3 and 4 lumbar vertebrae. The spinal tap to collect the CSF for examination is to be collected with aseptic precautions. The CSF will be collected in an amount of 3 to 5 milliliters into a labeled sterile container. The fluid to be collected at the rate of 4 to 5 drops per second. And transport the CSF to microbiology laboratory within 1 hour for culture and analysis. If that is not possible, inoculate the CSF into the TI medium. If TI medium is not available, incubate the CSF at 35 to 37 degrees Celsius with about 5% CO2 and store in an approved location. To collect the synovial fluid, after the skin around the joint is clean, the healthcare provider inserts a sterile needle through the skin and into the joint space. Fluid is then drawn through the needle into a sterile syringe. 3 samples are usually collected and are sent to different lab departments for analysis. Transport the specimen immediately to the laboratory. If the specimen cannot be examined immediately, fluid should be frozen and stored at negative 70% degrees Celsius until exam. Now to collect the pericardial fluid. A small needle is inserted to the chest between the ribs into the pericardium, and a small amount of fluid is withdrawn. And for transportation, a large volume should be submitted if tuberculosis infection is suspected. For cytology, add heparin or sodium citrate as an anticoagulant. Samples of the fluids are placed in various culture media in the laboratory, and the media is observed for growth of microorganisms. The test is performed when an infection of the heart is suspected or when the pericardial effusion is present. The pool of fluid is collected when the skin around the procedure site is clean. Local anesthetic is injected into the skin. A needle is placed through the skin and muscles of the chest wall into the space around the lungs, called the pleural space. The healthcare provider may use the ultrasound to find the best spot into the space and insert the needle. The fluid is then drawn out with the needle. The needle is removed and the area is bandaged. The fluid may be sent to a laboratory for testing. Lastly, the collection of peritoneal fluid is collected as follows. The puncture site will be cleaned and shaved if necessary. Local anesthetic is applied to the patient. The tap needle is inserted 1 to 2 inches into the abdomen. Sometimes a small cut is made to help insert the needle. The fluid is poured out into a syringe. The needle is removed. A dressing is placed on the puncture site. If a cut was made, no 1 or 2 stitches may be used to close it. Sometimes the ultrasound is used to guide the needle. There are 2 kinds of abdominal tabs. The first one is the diagnostic tab. A small amount of fluid is taken and sent to the laboratory for testing. The large volume tab, several liters may be removed to relieve abdominal pain and excessive fluid. And there are several criteria that can be considered by the laboratory on the basis which the processing of a specimen may not be done by the laboratory. Laboratory investigation of a sample are a waste of time and resources if following criteria are not fulfilled. And these criteria include missing or inadequate identifications, insufficient quantities, specimen collected in an inappropriate container, contamination is suspected, inappropriate transport or storage, or unknown time of delay.
Next, I will speak inoculations for blood culture. This page is ingredients of different blood culture bottles. The additions of adequate neutralized in blood culture bottles can help the growth of anaerobic bacteria. Additions of SPS can be used as anticoagulants. SPS can inhibit Neisseria species, Heterostreptococcus species, Gardnerella vaginalis, so we can add 1 to 1% gelatin to neutralize SPS. If the patient is taking antibiotics, we can use of blood culture bottles with ration. After culturing, if the blood culture bottle is positive and no foreign bacteria are identified, it is more likely to be contamination, such as coagulase negative Staphylococcus or Corynebacteria species. Terror can be seen if incubated in the 35 degrees Celsius incubator for 6 to 18 hours. Positive blood culture bottles. We can use the streaking technique to inoculate on the BAP, EMB, and CHO agar and incubate in an incubator of 35 degrees Celsius with 5% CO2. The inoculation of CDC and aerobic BAP should follow proper streaking methods and incubate in an 35 degrees Celsius and incubator. If we find it in grams, then we can inoculation to SDA as an addition. If bacterial growth cannot be detected with 5 days of continuous culturing, we can report a negative result. Next is inoculations for body fluids. If sample volume is more than 1 milliliter, we should centrifuge for 3000 rpm at 10 minutes and use a sterile loop to inoculate with selected agar pellets with the statement. If sample volume is less than 1 milliliter, we can use a sterile dropper and add 1 drop to inoculate the agar pellet. Use the three-way streak drawing method to inoculate on BAP, EMB, and CN agar. If the body fluid is subnephral fluid or pleural infusion, we should inoculate CHO agar as an addition because there might be a possibility of hemophilus species. Bacteria should not be found in body fluids, so any bacteria identified is politically significant. Next is an aerobic bacteria incubator, contains gas pack system, and aerobic glove box. Gas pack system is insulated agas are placed inside anaerobic containers along with a gas generator bag and indicate strip that is methylene blue and are cell completed in the presence of water chemicals present inside the bag which produce hydrogen and carbon dioxide gas. The produced hydrogen will react with oxygen present inside the gel thus producing water. If the gel container has water inside, methylene blue will change colors from blue to white, which help us to know the oxygen condition inside the gel container.
My part is pathogenic bacteria. This table shows the pathogenic bacteria in the blood. These bacteria can cause disease like bacteremia and sepsis. The types of bacteria have GPC such as Staphylococcus aureus, Streptococcus ehrlichia, and so on. The bacteria of GNC that can cause disease in blood are Neisseria meningitis and Moraxella catarrhalis. The bacteria of GPB are Listeria monocytogenes, Bacillus anthracis, Clostridium perfringens, and Clostridium difficile. The rest are GNB. The bacteria of GNB are Pseudomonas aeruginosa, Vibrio cholera, Acinetobacter baumannii, and so on. This table shows the pathogenic bacteria in body fluid. The disease cause depends on the location of the infection. They may cause meningitis, urinary tract infection, endocarditis, arthritis, and acute glomerulonephritis. The disease caused by each bacteria in body fluid are different. They may not only cause one disease, but also cause blood disease. Because there are many bacteria that can cause disease in body fluid, I take a few bacteria as an example. Here are the bacteria of GPC that can cause disease in body fluid. Streptococcus pneumonia can cause bacteremia and meningitis. Streptococcus ehrlichia can cause bacteremia, urinary tract infection, and meningitis. The rest bacteria of GPC can also cause body fluid disease. The bacteria of GPB that can cause disease in body fluid are Listeria monocytogenes, Bacillus anthracis, Nocardia, and Corynebacterium diphtheria. There are many bacteria of GNB can cause disease in body fluid, such as Klebsiella pneumonia, Escherichia coli, Proteus mirabilis, and Pseudomonas aeruginosa, and so on. Take a few bacteria as an example. Klebsiella pneumonia and Pseudomonas aeruginosa can cause urinary tract infection. Most of the bacteria in GNB cause urinary tract infection because most of them belongs to the Enterobacteriaceae. The bacteria of GNC are Neisseria meningitis and Moraxella catarrhalis. They can cause disease of blood and body fluid. Then the rest of the bacteria that can cause disease in body fluid are Borrelia and Chlamydia trachomatis. Borrelia can cause arthritis, and Chlamydia trachomatis cause urinary tract infection.
Now, let's introduce the bond media selection for plants. The first bacterium is Staphylococcus aureus. Staphylococcus aureus colonies appear as white colonies on 5% sheep blood agar and is beta-hemolytic. The bacteria has high salt tolerance on MSA agar, which can ferment mantle and produce yellow colonies, while the non-high salt tolerant bacteria will be inhibited and the one that does not ferment mantle, which produce pink or red colonies. Both triple plus pionics and triple purpose acolyte BAP is beta hemolytic, whilst triple purpose pneumonia BAP is alpha hemolytic. The next one is Neisseria meningitis. It is GNC. The bacterial colonies appear to be small, convex, gray, and sticky. Neisseria meningitis will have white colonies on CHO agar. The Muller-Hinton agar for Neisseria meningitis use 5% sheep blood based on CHO agar, with antibiotics to suppress flu spread. Enterococcal colonies are pinpoint smooth, entire colonies and can grow on CHO agar, but the colonies are not obvious on CHO agar. This slide talks about Moraxella catarrhalis and Haemophilus influenza. Moraxella can grow on CHO agar, and wagon wheel colonies can be seen on it. As for Haemophilus influenza, the bacteria can also grow on CHO agar and BHI agar with 3% FBA. The next one is Pseudomonas aeruginosa. The bacteria is beta-hemolytic on BAP. When the bacteria grow on nutrient agar, the colonies appear to be yellow. The bacteria can also grow on McConkey, EMB, and SOD agar. Vibrocorvus on TCBS agar produce large and yellow colonies because of the fermentation of sucrose. Vibroparahemolyticus. Hemolysis of Wakatsuma blood agar can be seen by extract of Kanagawa positive and negative strain of Vibroparahemolyticus culture on Wakatsuma blood agar. Yersinia piscis. The left picture shows that it can grow on blood agar after inoculation for 96 hours. The colonies on BAP are like poached eggs. Basilicus anthracis on BAP is gamma hemolytic. The colonies look like cotton. The next one is Listeria monocytogenes. The bacteria use selective media such as LPM, PALCAM agar, modified Oxford agar, CHRON agar, and ALOA agar. These agars are for Listeria only. Acinetobacter baumannii on BAP is beta-hemolytic. It also non-halophilic. Most strains cannot grow on 6% anesthesia or TCBS agar. Librovonoficus on TCBS can indicate that the bacteria is a sucrose mimicry. One of several media available for the selective isolation of Campylobacter is CCBA agar. The selective agar, CDSA, is recommended as a selective and differential medium for the primary isolation of Clostridium difficile from fecal specimens. Clostridium difficile spores. The spores are round-shaped and grow near the terminum. But in few circumstances, the spores will appear at the terminum. Bacteroides fragilis. There is a depression on the colony. It is an umbilical anabole. Gram-negative, rod-shaped bacterium. The colony appearance of Bacteroides fragilis on Ankylomycin lincomycin black blood agar is shown on left. The BBE agar is biplate, and the browning of the BBE medium is the result of exhaling hydrolysis. Clostridium perfringens can grow on CDC blood agar. There is a double-zone hemolysis on the agar. When the CDC blood agar plaque is examined under the light, colonies are surrounded by the double-zone hemolysis, which consists of inner clear zone and an outer hazy zone. The next bacteria is Fusobacterium necrophrome. Fusobacterium selective agar (FSA) is an enriched selective medium for the isolation and presumptive identification of Fusobacterium species. The used peptone is glucose agar can produce butyric acid.
And now I will introduce media selection for body fluid. This picture shows the pathogenic bacteria mentioned earlier, because the pathogenic bacteria in blood were mentioned before, so I will not introduce them in detail. Page start with an introduction to the pathogenic bacteria in UTI. The first is Staphylococcus saprophyticus. On the left is the pattern on BAP. You can see that this bacteria shows gamma hemolysis, and on the right is the growth pattern on MSA. You can see that it can grow on MSA but don't change color. This one is Staphylococcus duodenensis. You can see that it is showing beta hemolysis on BAP. It's Calibacia pneumonia. You can see that it shows gamma hemolysis on BAP and it appears pinker waxy on McConkey. The right-hand side is the EMB type, and then down E. coli. You can see that its colony on McConkey is also pink. Beta hemolysis on BAP and metallic green luster on EMB, followed by Proteus mirabilis. Obvious migration can be seen on BAP, and on the right-hand side is the shape on MHA. You can see the colony is white, and the button is the type on XLD. On XLD, you can see that the colony is a white dot inside a pink circle. The button is the last bacteria in UTI. Chlamydia trachomatis. Since chlamydia is an absolute intracellular parasite, it cannot grow in artificial laboratory cultural media, but needs to grow in living cells. Next is the common bacteria in meningitis, Streptococcus viridis. You can see that it shows alpha hemolysis on BAP, and the next is media. The growth pattern on BCYE median on the left-hand side. The BCYE is a special medium. Usually it is specially used to cultivate the gram-negative bacteria Leogenia pneumophilia. The medium is also used to identify Academobia carotides and to cultivate soil bacteria. The medium on the right-hand side is a plate culture of bacteria Nocardia astroides grown on 7H10 agar plates on 37 degrees Celsius. The kind of bacteria can grow on general bacteria, fungal, and mycobacteria media. Cloning appears after 4 days of cultivation and after 21 days of cultivation at 25 degrees Celsius. Colonies could be seen to produce pink to red mycelium covering the surface of the medium. The next one is the common bacteria in endocarditis. Corynebacterium difflurit. Beta hemolysis can be seen on BAP, and the one on the right is turret blood medium that specifically screens these bacteria. The next one is Borrelia species. The second cause of arthritis in Lyme disease. Because the cultural medium is not easy to obtain, the bacteria grow slowly and there are not many examples of successful culture, so culture is rarely carried out.
The next part, I'll introduce conventional methods of identification. First one, Staphylococcus aureus, GPC in cluster, catalase positive, coagulase positive, 1% glucose positive. The color will change yellow. DNA is positive, mannitol fermentation or MSA. The second one, Streptococcus pyogenes from group A, GPC in chain, strep test group A agglutination. You can observe the clumps formation immediately. PYR hydrolysis test positive. The third one, Streptococcus agalactiae from group B, GPC in chain, strep test group B agglutination, hippurate hydrolysis positive, CAMP test positive. Streptococcus pneumoniae, GPC in diploid, 6.5% NCL negative, SQE negative. Enterococcus species, GPC, catalase negative. Seldom of Enterococcus faecalis may be weak positive, while esculin positive. 6.5% NaCl positive, PYR hydrolysis positive, LAP positive. Neisseria meningitidis, GNC, catalase and oxidase are positive. Acid production from 1% glucose and maltose on CTA are positive. Lactose non-fermentation. Moraxella catarrhalis, GNC in diploid. Catalase and oxidase are positive. DNA is positive. Nitrate reduction positive. Butyrate esterase test positive. You can observe the development of a blue color during the 5-minute incubation period. Vibrio cholerae, sucrose fermentation. For TSIA tube showing A/A without gas and hydrogen sulfide production. If you use 0.5% sodium deoxycholate reagent, it will produce string reaction, but the specificity is low and it is rarely used now. There are two different biotypes for Vibrio cholerae, the classical biotype and the outdoor biotype. The classical biotype has CTX1 cholera toxin gene, while the outdoor biotype has CTX2 cholera toxin gene. Some basic characteristics and tests can differentiate between these two biotypes. The classical biotype has negative results for beta homolysis, RBC agglutination, VP test, and CAMP test, whereas the outdoor biotype has positive results for beta homolysis, RBC agglutination, VP test, and CAMP test. Vibrio parahemolyticus, GMV, halophilic, glucose fermentation, urease positive, H2S production. Pseudomonas aeruginosa, GMV in rods, oxidase positive, motile with polyflagella, It can produce water-soluble pigments which diffuse through the medium. The best known is pyothionein in blue-green color, which produced by a small proportion of strains. It is able to grow at 42 degrees Celsius. Biochemical testing panel. For TSIA appears K/K. This bacterium is only metabolizing peptones, so it's all red, with a gas formation and H2S production. Citrate positive, urease positive, motility positive. Actinobacter baumannii, GMB, glucose positive, citrate positive. It's able to grow at 42 degrees Celsius. ADH test positive. Clostridium difficile, anaerobic. It has endospore. Catalase negative. Luciferase negative. Lipase negative. Bacteroides fragilis, anaerobic. Catalase positive. Growth in 20% bile. Esculin hydrolysis positive. Alpha-fucosidase positive. Listeria monocytogenes, GPB, catalase positive, ChemTest positive, motility at 25 degrees Celsius. Its ability to grow at 4 degrees Celsius and in 10% NaCl. Bile-esculin and hippurate hydrolysis are positive. Methyl red and VP test are positive. Bacillus emphasis, GPB, catalase positive, pearl string test positive. Yersinia pestis. For TSIA appears K/A. It ferments glucose only, with a gas and hydrogen sulfide production. LNPG test positive. Nitrate reduction positive. Motility at 25 degrees Celsius. Urease test positive. It can grow at 4 degrees Celsius. The last but not least, Vibrio bonificus, GMB, oxidase positive, motility positive, lactose non-fermentation. It is classified into two biotypes according to some biochemical tests. The biotype 1 has positive results for indole test, ornithine test, and mantle fermentation, whereas the biotype 2 has negative results for these tests.
Campylobacter jejuni, catalase positive, oxidase positive, motility positive. Hippurate hydrolysis positive, nitrate reduction positive, intazole acetate hydrolysis positive. Haemophilus influenza, GNCB, glucofermentation, catalase positive, oxidase positive. Can use satellitism test. XV vector requirement. X vector means heme and V vector means NAD. H influenza need X vector and V vector. Porphyrin test. Porphyrin test is used for rapidly detecting if Haemophilus species requirement factor X to grow, enzyme which convert ALA to porphyrin in the biosynthesis for X vector are produced by Haemophilus parainfluenza. UV light can help us to distinguish Haemophilus parainfluenza and Haemophilus influenza. If it do not have red appear, means it is Haemophilus influenza. Clostridium perfringens, anaerobic, GPAB, have endospore. Alpha toxin, lipase, negative. Lactose fermentation, reverse CAMP test, positive. Fusobacterium necrophone, anaerobic GNAB, Indo-positive, lipase negative, Staphylococcus saprophyticus, GPC, glucose positive, catalase positive, coagulase negative, gamma hemolysis, Streptococcus viridans, GPC, glucose negative, catalase positive, alpha hemolysis or gamma hemolysis, oxidase negative, Streptococcus leukodenicis, GPC, catalase positive, coagulate test. If you test by tube, it will negative. It shows positive on the control. Corynebacterium diphtheriae, gram state GPB, non-motility, coagulate negative. Nocardia GPB, catalase positive. Strike aerobes. Some species are particularly act fast, mean a less concentrated solution for sulfuric or hydrochloric acid should be used during the standing produce. Campylobacter fidus, GMB, catalase positive, oxidase positive, motility positive. Afraid to cold. To best control above 30 to 42. Borrelia species must be viewed using dark microscopy. Most species are obligate, anaerobics. This result is not easy. Use gram stain, catalase negative, oxidase positive or negative. Chlamydia, trachomatis only can use cell control. Klebsiella pneumoniae, GMB, glucose fermentation, lactose fermentation, oxidase negative, nitrate reduction positive, catalase positive. Gas production, H2S positive, negative, citrate positive, BP positive, urea positive, Indo negative, motility negative, IPA negative, ORN negative, LYS positive. E. coli, GNB, glucose fermentation, oxidase negative. Gas produced, NH2S negative, citrate negative, VP negative, urea negative, Indo positive, motility positive, IPA negative, ORN unable to grow, LYS positive. Proteus mirabilis, GNB, glucose fermentation, oxidase negative. Gas production, H2S positive, citrate unable to grow, VP unable to grow, urea positive, Indo unable to grow, motility positive, IPA positive, ORN positive, LYS negative.
The next part, I'm going to talk about AST. First, about 15 minutes rule. We use a single cannoning from non-selective agar to prepare inoculant suspension. Standardized inoculant suspension to morphine length 0.5 and inoculate to agar plate in 15 minutes. Cover the antimicrobial disc in 15 minutes. Finally, put into incubator in 15 minutes. This is the plate you inoculate without any gap. Should look like this, not like this. For blood and body fluid specimens. First, for blood specimens. AST isn't required for specimens that was suspected to be contaminated, but we have to preserve the specimens. For Staphylococcus species, when you use broad dilution method, we need to add 2.2% NaCl for oxazine, and the table is for Staphylococcus species. It can help us to identify susceptible or resistance. For Streptococcus species beta-hemolytic group, we need to use Hinton agar with 5% sheep blood. For Streptococcus pneumonia, we use Hinton agar with 5% sheep blood. And this is for Enterococcus species, Neisseria meningitis. We use Hinton agar with 5% sheep blood in 5% CO2 incubator. And this is for non-enterobacterials, and this is for Pseudomonas aeruginosa, a Ginetobacter species. For anaerobies, they need heme, vitamin, and leg sheep blood to alive, so we add those vectors to both or agar and put in a anaerobial incubator. For Listeria monocytogenes, we can use those medicines to treat patients for Haemophilus influenza. We use HTM with 5% CO2 incubate 16 to 18 hours. And this table is for Enterobacterials. And I'm going to introduce the automatic AST. The VTEC has 3 advantages. First, an accurate microbiome identification, and to test antibiotic susceptibility testing. We how to operate the steps. Now I will tell you. First, we find the blood specimen culture flag positive and transfer 1 ml blood to 1.5 ml Ependorf and add 200 microliter lysis buffer and both test for 10 seconds and centrifuge for 2 minutes at 13,000 rpm and discard the supernatant and washing the pellet in 1 milliliter of washing buffer and centrifuge for 2 minutes at 13,000 rpm and discard the supernatant, then suspend the pellet in 500 microliter of sterile monosaccharine and stand down light to morpho length 0.5. Finally, set up the machine, and this is our work division. Thank you for your attention.